Hello friends, welcome back. In this session, I shall be discussing on histology of male reproductive system. This is diagrammatic representation of male reproductive system. This is testis, epididymis, which are located in scrotal bag. The tail of the epididymis continues as vas deferens, which enters the abdominal cavity through inguinal canal, then passes down into pelvic cavity. It is related posterior to the urinary bladder. Ductus deferens, after joining with the duct of seminal vesicle, forms ejaculatory duct. There are two ejaculatory ducts on either sides. They pierce the prostate and empty themselves into that part of the urethra which is within prostate, that is prostatic part of urethra. Then through the penile part of the urethra it finds exterior. Learning objectives are describing the microscopic structure of testis, epididymis, vas deferens and prostate. This picture shows testis and its interior. Coverings of the testis have been removed. Tunica vaginalis can be seen here. This white colored one is tunica albuginea, which sends connective tissue septations inside the core of the testis towards the mediastinum testis, dividing it into 200 to 300 lobules. Each lobule has convoluted tubules, 2 to 3 convoluted tubules located within the lobule, which both ends of the tubes open towards the rete testis. Rete testis receives the collections from the tubules and sends it to the efferent ductules to the head of the epididymis. This is epididymis head, this is body. Epididymis is a single tube convoluted on itself very highly, and this is the tail of the epididymis, and this is ductus deferens. Interestingly, testis is a gland which secretes cells. They are gametes. It secretes spermatozoa. Testis is also an endocrine gland which secretes testosterone. Let us look at one such lobule, how the seminiferous tubules are located within the lobule of testis. This is how one lobule of the testis shows highly convoluted seminiferous tubules. These are loop-like tubules. Both the open ends are open towards the mediastinum testis or towards the rete testis. So it is highly packed lobule with seminiferous tubules. When we take transverse section through the testis for histological preparation and stain it with H&E, we observe something like this. So all these are seminiferous tubules which are cut in different tangents, transversely, obliquely, longitudinally. We find many such waves. All these are seminiferous tubules. And in between the seminiferous tubules, there is some space which is occupied by delicate connective tissue and also some of the cells. So this space is interstitial space in between the seminiferous tubules. So this is the high power view of one seminiferous tubule, so which shows a special stratified epithelium. We find many cells in strata, so this is a special variety of stratified epithelium. This is actually harboring all the cells of spermatogenesis, spermatogonium till matured spermatozoa. So all of them are here which are maturing. Spermatogenesis is taking place in these tubules. And in between the tubules, this is interstitial space, and we find some large cells. These are called Leydig cells which secrete testosterone. Let us understand by this picture how cells of spermatogenesis are located within seminiferous tubule. The same, the testis, one such seminiferous tubule has been elongated here and this is magnified, and this part of the tube, wall of the tube has been cut and it is magnified here. This is the wall of the tube which shows muscle-like cells. They are called myoid cells, and all these are cells of spermatogenesis. The basal cells are here. These are spermatogonium, which are on the basal side. Then they differentiate into these are primary spermatocytes. The largest cells which are seen within the thickness of the seminiferous tubule is primary spermatocyte, which divides by meiosis 1 to form secondary spermatocytes, which are haploid in number, and then spermatids and spermatozoa. So all the process of spermatogenesis and spermiogenesis are happening within the tube. Interestingly, you observe one thing here. There are some yellow colored cells which are extending all over the thickness and which are pocketing these cells of spermatogenesis on their surfaces. So these yellow colored cells in this picture are Sertoli cells. Let us understand how these Sertoli cells are actually related to all these things. This is three-dimensional diagrammatic representation of seminiferous tubule, and its wall has been enlarged here. This is wall of the seminiferous tubule, and this cream colored cell is Sertoli cell. They extend from base till the lumen. But on the surface of the Sertoli cell, all the cells of spermatogenesis are packed. They are not within the Sertoli cell, but on the surface of the Sertoli cell, they are present. They are hidden, or they are uh, mm, pushing themselves into the Sertoli cell cytoplasm, taking its cell membrane. This is a primary spermatocyte and this is secondary spermatocyte. This is after meiosis 1, that is, this is haploid. Primary spermatocyte is diploid. And after spermiogenesis, the mature spermatozoa will form, the head of which will be uh, pushed or hidden inside the within the wall of Sertoli cell, and tail is projected towards the lumen. This is how Sertoli cell is giving nutrition to these cells. It is producing a Mullerian regression factor. It is producing androgen binding protein. It is nourishing all these cells of spermatogenesis. After understanding how these Sertoli cells are related to cells of spermatogenesis, let us again look at this in diagrammatic representation. This is wall of the seminiferous tubule, and the yellow colored cytoplasm is of Sertoli cell. And at the base, we find spermatogonial cells, then primary spermatocytes. Then they divide meiotically into secondary spermatocytes, then spermatids and spermatozoa. In realistic picture, we can observe here. So this is wall of the seminiferous tubule, which has smooth muscles or myoid cells, which help in squeezing out the contents uh, quickly and releasing the spermatozoa into the lumen of seminiferous tubules and help in motility towards the distal part of the tubule. And these are Sertoli cell nuclei. We cannot define the outline of Sertoli cell, but nuclei are all are mixed here, and it appears a special variety of stratified epithelium. Sertoli cells, gonial cells, primary, secondary spermatocytes, spermatids, and spermatozoa all are adding up to the number of nuclei. So all these things are observed here. And outside to the tube, within the interstitial space, we find large cells with the dark nuclei, round nuclei. These are Leydig cells, which secrete testosterone. Along with these interstitial cells, we find capillaries. Capillaries indeed are covering all over the surface of these seminiferous tubules. So that itself will make tunica vasculosa, which was not clearly observed in gross anatomy. This is how in low power view testis appears. It has seminiferous tubules, and epithelium of the tubule is special variety of stratified epithelium. The nuclei here are Sertoli cells, gonial cells, primary, secondary spermatocytes, spermatids, and spermatozoa. You can see the tails of the spermatozoa making swirl pole here within the lumen, and these are interstitial cells. This is one more picture. You can see the tails of the spermatozoa, how they are making swirl pole. All these are the nuclei of spermatids and spermatozoa. Basally, these are gonial cells. We can observe myoid cells or muscles-like cells in the wall, and these are Leydig cells, interstitial cells, and these are capillaries. You can also observe the capillaries all around with RBCs filled in it. That is tunica vasculosa, around the seminiferous tubules. Epididymis is here. It has head, body, and tail. Indeed, the body and tail of the epididymis is a single tube which is highly convoluted on itself. It's a single tube. It receives all the contents from seminiferous tubules through the network of tubes in rete, that is rete testis, and through the efferent ductules into the head part. And then this single tube conveys all the contents through it to the ductus deferens. So when we take section through the epididymis, that will also show, just like testis, the ducts of epididymis, the duct of epididymis, that is single tube, cut at different duct of epididymis. When cut, it appears something like this. A single duct which is highly convoluted is cut at different tangents. It shows many different shapes like circular, U-shaped, J-shaped, C-shaped, depending on the section how it is going. This is obliquely cut. This is longitudinally cut, transversely cut. In higher magnification, it appears like this. This tube or duct of epididymis is lined by epithelium. This is pseudo-stratified ciliated epithelium. So cilia are here. Pseudo-stratification is because of multilayer appearance, but all the cells of epithelium are touching the basement membrane. There are some principal cells which are long and basal cells which are at the basal side. They give stratified appearance. These are columnar cells with cilia, stereocilia. They are long microvilli like stereocilia. They are not motile cilia. The wall is also made up of smooth muscles. And all this is interstitial connective tissue in between the duct. One more picture shows stereocilia and pseudostratified ciliated columnar epithelium, smooth muscles in the wall, and we can observe many of the times contents within the duct, that is spermatozoa. It can be present or it may be absent. Just go by the epithelium, that is pseudostratified ciliated columnar epithelium. These are stereocilia. They are non-motile cilia. This is one more picture. I have intentionally placed it here to show if the section is going through the wall of the duct, then it appears something like this. At the levels of the nuclei, the section is going like this. That's why it appears as if the tube has something. This also. So such pictures, if you find, just think that the section is going through the wall. This is one more picture. This is a duct of epididymis. These are smooth muscles, pseudostratified ciliated columnar epithelium. These are stereocilia. This is the content, the magnified view. In the epithelium, we find some principal cells and some basal cells. Here, both Slides of testis and epididymis have been shown. These are all seminiferous tubules. These are all duct of epididymis. The lining epithelium of seminiferous tubule is stratified in appearance. So it has all the cells of spermatogenesis with Sertoli cells. Wall is filled with muscle tissue, and there are interstitial cells of Leydig with capillaries around. In duct of epididymis, it is lined by pseudo-stratified columnar epithelium with stereocilia. The wall is having muscle tissue. It may be filled with content or may not be filled with content. The function of seminiferous tubule is to produce spermatozoa. The function of epididymis is to give partial maturation to the spermatozoa and also to store the spermatozoa. This is transverse section of vas deferens. Look at the diameter of the lumen and thickness of the wall. The ratio between the lumen diameter and thickness of the wall is very less in this tube. This is the only tube in the body which has thickest wall compared to lumen diameter. In the wall, it has smooth muscles, inner and outer longitudinally arranged smooth muscles, and in the middle it has circularly arranged smooth muscles. It is lined by epithelium, that is pseudostratified columnar epithelium with stereocilia, just like epididymis. This is one more picture showing the lumen, epithelium, and its wall. It has its own artery, that is artery of ductus deferens. If it is in the section, it can be seen. This is higher magnification of the lumen of vas deferens, where appreciation can be done for lining epithelium. This is pseudo-stratified columnar epithelium with stereocilia. The passage of contents through the ductus deferens from tail of the epididymis till the ejaculatory duct is called emission. From the ejaculatory duct through the urethra out is called ejaculation. Prostate is accessory gland of male reproductive system. It is located below the urinary bladder at the neck around the initial part of urethra. The urethra passes through the prostate, that is prostatic part of the urethra. This shows coronal section through the prostate, where this blue colored one is posterior wall of the prostatic part of urethra. It has raised elevation called seminal colliculus. On either sides, you can see these two openings. These are openings of ejaculatory duct. This is how contents from the testis through the epididymis through the ductus deferens reach the prostatic part of the urethra after getting secretions from seminal vesicle through ejaculatory duct. Openings of ejaculatory ducts are present on seminal colliculus. Prostate is also having many glands within its core. All of them are opening separately by many ducts in the posterior wall of prostatic part of urethra. This is again an exocrine gland. So this glandular tree is within the mass of prostate. So all this glandular part of the prostate is separated by stroma of the prostate that is formed by fibrous tissue and muscular tissue. This is section through the prostate where urethra can be seen here lined by transitional epithelium, that is prostatic part of the urethra. Urethra can be there in this section or it may not be there in this section. Looking at the tissue of prostate, identification has to happen. So all this is stroma of the prostate, and these are glandular parts of the prostate, that is parenchyma. In this picture, we can observe there is pink colored stroma, that is fibrous and smooth muscular tissue, and all this is glandular part, acini of glands. This is glandular epithelium. Epithelium is lined by simple cuboidal to columnar variety. It secretes, all the secretions will reach to the posterior wall of the prostatic urethra by separate openings. This is low power view with prostatic urethra. This is high power view. Prostate is a fibromusculoglandular structure. So these are low power and high power view of the same. Stroma is made up of fibromuscular tissue. Glandular part is made up of glandular epithelium. So glandular epithelium, this is fibromuscular tissue. In old prostates, the secretions within the glandular tissue can concrete and form corpora malacia. These are eosinophilic bodies, precipitation of the secretions of the prostate. Prostate contributes to the seminal fluid nearly second to seminal vesicle. So I shall give you the composition of seminal fluid and what is secreted and how much is secreted by seminal vesicle, testis and prostate at the end. This ends the session. Thank you.